So let's begin with this third competency, that is your nerve muscle physiology. When I talk about nerve muscle physiology, it is a wholesome process, I would like to tell you, because for a muscle to act, you need the nerve conduction, you need a signal for the muscle, you need excitation to the muscle to contract it. So what is the basis? How does this entire process occurs? We are going to start with the muscle followed by the nerve. When you take a muscle cells, they are also like our nerve fibers. They are also going to be excitable. They are going to be responding to the stimulus by producing action potential, like we discussed in general physiology. There is a resting membrane potential for your muscles too. Can anyone tell me what is the resting membrane potential for the muscles? Yes, just have this as a question. We are going to discuss that again. You will be having an entire structure that is going to be like dissected out one by one. You have a big muscle fibers. That muscle fibers is packed actually. How does it packed? Everything we are going to discuss in this chapter. Basically your muscles are classified as skeletal muscles, smooth muscles, and cardiac muscles. So first what we are going to discuss is your skeletal muscle. Skeletal muscles are striated. One of the very important clinching point is striations. And they are voluntary in nature, whereas your smooth muscles are going to be autonomous in nature. So this voluntary skeletal muscle are going to be supplied by the nerve that is going to be controlled, that is going to be having a controlled action or volunteered action. Is it clear? So what it is, let's discuss. To start with, a skeletal muscle cells is otherwise called as myocyte. We are calling it as a muscle fiber. We call it as a myofiber, etc. Your skeletal muscles never are homogeneous. They have got several dipping, invagination into your cytoplasm. These invagination, you call it as T-tubules. You're going to call these invaginations as T-tubules. So your muscles are not going to be homogeneous, your skeletal muscle fibers. They are going to have a dipping or invagination into cytoplasm. You call it as T-tubule. And they are going to be having a covering. Your entire cell of your skeletal muscle are going to be in a covered manner. What is that covering called as? You call it as a sarcolemma, or the cell membrane that is going to be covering is called as sarcolemma. As you can see, at the end of the dipping, you're going to have a numerous foldings, isn't it? So can you see in the right side end, can see numerous foldings. Yeah, on this folding point, you will be having the neuron that is going to coming and attach it, so that there is a signal transmission. This place is called as motor end plate. This place is called as motor end plate. So between your motor end plate and your neuron, you're going to have a gap, isn't it? That gap is called as neuromuscular junction. Now coming back, you're going to have in between the T-tubule, in between that invagination, you have a bulky organelle, isn't it? This bulky organelle is called as sarcoplasmic reticulum. This bulky organelle is called as sarcoplasmic reticulum. As well, you can see a small muscle, there is a organelle, long slender organelle. What is that? That is called as your myofibrils. That is called as your myofibrils. Now, let me take you into the depth of myofibril at this point of time itself. In myofibril, you could see some lines, isn't it? So, what is this lines called as? These lines are called as Z line. These lines are called as Z line. And between one Z line and other Z line, you have a gap, isn't it? This gap is called as sarcomere. This gap is called as sarcomere. Sarcomere. Now understand, this sarcomere is the functional unit of a skeletal muscle. This sarcomere is a functional unit of skeletal muscle. You can see this sarcomere under microscope. So you can see this sarcomere under microscope. Okay, now let us quickly go through some one-liners. The neuronal endings are going to synapse with a muscle only at the motor end plate. The sarcoplasmic reticulum has a unique function because They are going to store your calcium. They are going to store your calcium because this calcium is essential for the contraction of your skeletal muscle. Coming back to the myofibril, this is going to be the one that have got thick filament, thin filament, and the sarcomycin. So we are going to discuss what is thick filament, we are going to discuss what is thin filament, and we are going to discuss what is sarcomycin. Before moving to that, every skeletal muscle has got 9 to 100 myofibrils. So you have a skeletal muscle cell. These muscle cells are going to be containing 9 to 100 myofibrils. And the resting membrane potential of a skeletal muscle is going to be minus 90 millivolt. As I told you already in your general physiology, they are going to be depolarized by sodium channels. They are going to be excited by sodium influx. So sodium influx, which is gated by the voltage gated sodium channels, which are going to be opening at a sub threshold, that is minus 55 millivolt. Both, okay, this is going to be sodium influx. But when will your calcium is going to get influxed? When will your voltage gated calcium channels in the skeletal muscle are going to be opening? That will be at the plus 40 millivolt. So why is this important? Because at this point of time, there is going to be a contraction of your skeletal muscle that is at plus 40 millivolt. And exocytosis of neuron is going to be occurring at this plus 40 millivolt. But remember one important thing, in a smooth muscle cell, you don't need a sodium influx. You can no need a sodium influx, but your calcium influx itself is sufficient for the excitation of your smooth muscle cells. That is what we will be discussing it later. Now coming back to the functional unit of a skeletal muscle, that is called as sarcomere. So this is going to be your sarcomere. This sarcomere is going to be guarded by Z line. This sarcomere is going to be guarded by one important thing, that is going to be Z line. Now you can see that for a skeletal muscle to get contracted, you have to have a contraction of your muscle fibers. So the muscle fibers are going to be arranged by numerous myofibrils. Each myofibril contains numerous sarcomeres, and this sarcomere is going to contain very important thing, that is actin, myosin, titin, and various other muscle protein. So what we are going to do is we are going to discuss these muscle protein. Before I move towards the muscle protein, this is what I exactly told you. So there is a tendon that is covered by epimysium. You can see inside epimysium, you have various fascicles. So these fascicles are wrapped by perimysium. These fascicles contains numerous muscle fibers, and these muscle fibers are going to be covered by endomysium. These muscle fibers are covered by endomysium. Okay, so these endomysium inside your muscle fibers, inside this muscle fibers, once again I repeat, there are going to be numerous myofibrils. These myofibrils contain sarcomere, and this sarcomere is a place where actin myosin interacts. Okay, that is what we are going to discuss. Sarcolemma, I told you, right? So these are the cell membrane of your muscle fibers. This sarcolemma, okay, it is going to consist of cell membrane that have an outer coat of polysaccharides and a collagen fibrils. They are going to be also having a collagen fibrils. Remember, sorry for the disturbance for my screen. I'll just let it come back. I'll just put back my screen. Before that, I like to add upon that. So once you understand that there are going to be numerous myofibrils, what happens to these myofibrils? These myofibrils are nothing but they are going to have a sarcomere, isn't it? That sarcomere is going to be having an actin myosin interaction. Then I call it as actin myosin interaction. How I'm going to call it as a thick filament and thin filament. Remember, your thin filament is made by the tail of your myosin. Your tail of your myosin is the one that is going to prepare what? Your thick filament, and you're going to have another one that is called as thin filament. This thin filament is nothing but made up of remaining proteins that are actin. You have a troponin, tropomyosin. All these things are going to make up. Then you have the sarcoplasmic reticulum, as I already told you. Then you have the sarcoplasm. What is the plasm? Literally means it is a cytoplasm. They are going to provide the nutrient So this sarcoplasm is going to consist of a mitochondria, which is going to act as a powerhouse of your skeletal muscle. Okay, so now coming back, I would like to draw this. Okay, you can just draw along with me. So what happens is a sarcomere is going to be consisting of two Z lines. So what I'm going to draw is I'm going to draw two Z lines. So this is going to be your Z lines. And what we are telling is, so between the skeletal sarcomere, okay, so you have Z line, isn't it? Between these two Z line, there is going to be another line that you call it as M line. That you're going to call it as M line. M line is nothing but it is going to be the center line. M line is nothing but it is going to be the center line. Okay, so now you know that between the two Z line, there is going to be one central line that is called as M line. Now what happens is, okay, you're going to have a thick filament and a thin filament. Okay, where does this thick filament will be there? Where is the thin filament? Your thick filament actually will be like this. Okay, your thick filament will be like this. Okay, so you're going to have a thick filament at the center point. I can draw like this. So this is going to be your thick filament. So this is called as your thick filament. Okay, thick filament. Okay, so this is going to be your Z line. This is going to be your Z line, and this is going to be your, this is going to be your M line. This is going to be your M line, and this is going to be your Z line. Okay, between two Z lines, you're going to have a M line, and in this M line, there is going to be a thick filament. So the zone where there is a thick filament, you call it as a H zone. So the zone where there is going to be a thick filament, you call it as a H zone. So the region where A band, there is only thick filament, that is called as a H zone. What is A zone then? The region spanning from one end of the thick filament to the other end of the thick filament, you call it as a zone. So what is this zone called as? This zone is called as A zone. Clear? So this is going to be called as A zone. Okay, now you understood what is A zone, isn't it? Now let's move on towards the next one. That is going to be your, okay, what is that? Thin filament. Where is your thin filament located? Your thin filaments are actually located over your Z line. I can just show you. So where does your thick filament present? Your thick filament is present over the M line, whereas your thin filament are going to be present over your Z line like this. So this is going to be called as your Z line. So this is going to be called as your Z line. Now understand. So what happens is there is going to be interaction. Actually, can you see here? So this is going to be extending into your, this is going to be extending into your A zone. So A zone kulla ungalku in a thin filament extend agudu. So how does your thick filament present? So you saw already. How is your thick filament present? So it's going to be present like this. Can just draw this and keep in your notes so that it won't get confused for you. Okay, so like this you have. So this is going to be your thick filament, and this zone is going to be your thin filament. So this is going to be your thin filament. This is going to be your thick filament. Clear? So now you understood what is thick filament, what is thin filament. You understand what is A zone. Between the region within A band. So this is going to be called as A band. So this is going to be called as A band. So that is your thick filament. Okay, so this thick filament is A band is going to be from one end of the thick filament to the other end of the thick filament. And what is H zone? This zone is called as H zone. That is the region within A band where only your thick filament is present. So can you see here? So here only your thick filament is present. Here only your thick filament is present. This zone is called as A zone. So I'll again show you. So here you can see this region where there is going to be only your thick filament is going to be present. So this region, there is only a thick filament is present, no thin filament is present. So the region where only thick filament is present, the region between the Z internal line, internal ends of the thin filament. So this is going to be the internal end of your thin filament. Between the two internal end of thin filament, where only thick filament is present, that is called as edge zone. So now you understand what is A band. A band is between the two thick filament. Edge zone is between the two thick filament, where there is no thin filament at all, because your thick filament extends into your thin filament extends to your thick filament. So there is an interaction. So the place where only thick filament is present, that is called as a edge zone. Now coming back to what is a C zone then. So you understood what is a band. You understand what is the thick filament, that is your edge zone. Now what is C zone? C zone is where the region possible where there is going to be interaction between your thick filament and thin filament. Can you understand? See here. So this is the place where there is going to be interaction between your thick filament and your thin filament. So this is the place where there is going to be interaction between your thick filament and thin filament. This interactive place is there, right? This place of interaction, you're going to call it as, this place of interaction, you're going to call it as C zone. Okay, so now first what you saw, edge zone, then what you're going to see, C zone. Then what is the I band? The I band is nothing but you have the end of A band, isn't it? So the end of A band, what is A band? A band end means the end of the thick filament. Okay, between the end of thick filament, okay, between the end of thick filament, where is your thick filament? So here, this is where your thick filament ends, and this is where your Z line is there. So this place is exclusively going to have thin filament alone. There is no thick filament. So the end of thick filament to your Z line, you call it as you're going to call it as a I band. You're going to call it as a I band. Okay. So now, now what are the things again? I will repeat from one. Number one, first you saw two Z line. Between the two Z line, you're going to put at a central place. You're going to put a line that is called as M line. So first one is a Z line. Second one, M line. Third one, what you're going to do is you're going to have a thick filament over your M line. You're going to have a thick filament over your M line, and you're going to have thin filament over your Z line. So it is going to be arranged like a band. Now, the band, the place, okay, where your thick filaments are there completely. So the place where your thick filaments are there, from one end of the thick filament to the other end of thick filament, you call it as a band. Number 3. Okay, number 4, we are going to have a band. But what happens is your A band is going to be overlapping with your thin filament, isn't it? So the place where there is no overlapping of your thin filament, you're going to call it as a edge zone. Number 4. Number 5, you have this interaction place, isn't it? The interaction between the thin filament and your thick filament, that region is called as C zone. And then your purely your thin filament, isn't it? So that means from the ending of your thick filament to the Z line, you're going to have a region, isn't it? That region is called as I band. Okay. This is what you need to understand. I hope you can understand this. Okay. Once you understand this, next, okay, what you have to know is between the two M zone, M line and your Z line, between your M line and your Z line, there should be a, okay, protein that is going to be connecting your Z line and the M line, because for the proper interaction, you need a proper coordination between your M line and Z line, and this coordination is given by the protein called as titin. It is given by the protein called as titin. Okay, what this titin is known as? This titin is called as muscle spring. This titin is called as muscle spring. Why do you call it as a muscle spring? Because they are going to be responsible for elasticity of the muscle. Remember, if a MCQ is asked, what is the protein that is responsible for the muscle elasticity? Then you're going to name a muscle, name the titin. Then you're going to name the tighten. Is it clear? Now, okay, this is going to, what it is going to do? This is going to connect. So what this is going to do? This is going to connect. Okay, this is going to be connecting your what? M line and a Z line, thus providing scaffolding of the sarcomere. Clear? Are you clear? Next, you are going to move for one intermediate filament of the muscle. What it does is, this connects your Z line. You're going to connect your Z line to the plasma membrane. So you have a plasma membrane, right? Okay, so that is going to be connected with the cell membrane, and the one that connects your cell membrane with the Z line, that is called as desmin. That is called as desmin. So this desmin is going to connect your Z membrane, Z line to your plasma membrane. Then what does your actinin going to do? Actinin is another structural protein that binds the actin to your Z line. So you have actin myosin 2 protein. I'm going to discuss it now. Before moving to that, you have actin. This actin is going to be connected to the Z line by a protein, okay, by a protein that is called as, okay, that is called as actin in, okay, actin in. So that is called as actinin. Okay, now you have another one, that is dystrophin glycoprotein complex. So I'm discussing 4 structural protein here. Number 1, before moving towards actin, myosin, I'm discussing 4 structural protein. Number 1, titin. Number 2, desmin. Number 3, actinin. And number 4 is your dystrophin glycoprotein complex. Dystrophin glycoprotein complex. Dystrophin glycoprotein complex. So what it does is, this is going to add a strength to the muscle by providing a scaffolding of the fibrils and connect them to the exterior. So what are the component of dystrophin glycoprotein complex? There is going to be dystroglycan, there is going to be merosin, syntropins, and sarcoglycan, and it's going to consist of sarcoglycans. So now once again, what is A band? A band is a region that is going to be connecting from one thick filament, region between one thick filament, okay, one thick filament, okay, end to other thick filament, end to other end of the thick filament, other end of thick filament. So that is going to be called as A band. Number 2, what is S zone? The region between the, okay, region which is going to be within the A band, where only thick filament are going to be present, where only thick filament is going to be present. Thick filament is going to be present. Okay, what is C zone? C zone is nothing but the region, okay, of possible actin myosin interaction. Possible actin myosin interaction, or otherwise I can call it as the region where there is going to be an interaction between the, your actin is otherwise thin filament and myosin is thick filament interaction. Thick filament interaction. Okay, what is I band? I band is the region between external end of A band and your till your Z line. Okay, so what is M and S Z line? M and Z line are connected by titin. Titin connects M and Z line. These are the 5 things I want you to remember, guys. This is going to be the take home message. This is going to be the take home message. Now coming back towards this. Okay, so there is going to be a thick chain, there is going to be a myosin like this. So you have a thick filament. So you have a edge zone. From edge zone, I'm just removing the thick filament. So this is going to be your thick filament. This is going to be your M line. So thick filament, okay, you know titin. What is the role of titin? It connects M line to Z line. Can you see here? This is your titin. And what happens inside the thick filament? You can see some yed. Okay, so these thick filaments are made up of myosin. Your thick filament is made up of tail of myosin. This is going to be a tail, isn't it? So this tail of myosin is what forms a structure of your thick filament. Okay, the tail of myosin, if it is there is going to be yet part. What is the role of your yet part? This yet part is going to be the contractile This is going to be the contractile part of your myosin. It is going to be the contractile part of your myosin. Okay, so this is going to be the tail of myosin, and you have a contractile part, that is your head of myosin. This head is going to have a binding site for the actin. Your tail forms the body of thick filament. You have a neck and head that is going to be popping out. The popping out part is going to have a binding site for actin. Okay, then what is your thin filament made of? Your thin filament are going to be made of. Can you see here? So this is going to be your Z line. From the Z line, you have actinin. Okay, you have actins. So there is going to be three important bonds which is going to be making up your thin filament. Number one, actin. Number two, tropomyosin. Number three, troponin. Can you see here? So you have a troponin. You're going to have a tropomyosin. So when you're going to see the if you're going to zoom in and see, you're going to have an actin. You're going to have a troponin, you're going to have tropomyosin, and apart from it, you also have nebulin. Which we will be discussing. Now, coming to the actins, these actins are going to be a monomer. So, G-actin we call it as. These G-actins have to fuse together. Can you see here? These G-actins, so these are the individual actins. So these G-actins fuses together to form a chain, and that is called as F-actin filaments. This is called as F-actin filaments. So in order to have a filamentous actin, what you need? In order to have a filamentous actin monomers has to be binded. How it is binded? They are going to bind by three important molecules. I can call it like this. So when you're going to have an actin like this, okay, can you see here? So this is going to be one actin monomer. And what happens is, I'll just draw you and show you. Okay, so you're going to have one actin like this. This has to be moving like this. Okay, can you see here? So this is going to be the other actin molecule. It's going to be coming. I am poor in drawing anyway. So this is going to be it looks like at least like this, it will be looking. Okay, it is not uniform, but again, it's going to be like this. So you have an actin, isn't it? So for this actin, okay, for this actin to be together, you have three other molecules. 1, 2, and 3. What are the three molecules? I call it as CIT. CIT. What is the CIT? Troponin C, troponin I, and troponin T. So these are the regulatory proteins that are going to regulate your muscle contraction and relaxation. Number 1, troponin C. It is going to contain the binding site for calcium that initiate your contraction. So C for calcium, remember, trop C. C for calcium, it is going to be helping in the binding site of calcium and initiate your contraction. Then you are going to have a trop I. What does I stands for? I stands for, so it's very thin, isn't it? So what is your thin filament made of? Actin. So remember, it is going to be the actin place where it inhibits the interaction between your myosin and your actin. So between your myosin and actin, it prevents the interaction. And what is going to be your trop T? Remember, trop T is going to be binds the troponin component of your tropomyosin. You're going to have other protein, isn't it? That is tropomyosin. So your trop T is going to bind with tropomyosin. It is going to bind with tropomyosin. So these are the long filaments located in the groove between the two chains of the actin. Can you see here? So can you note here? So between the two component of your actin, there is a thin filament that is called as tropomyosin. Now I guess you know what is troponin, what are the different troponins present, what is tropomyosin, etc. Okay, now what happens is this tropomyosin, I told you, right? These tropomyosin are going to be acting like a covering. They are going to be acting like a covering that is going to cover your. So this is going to, what happens is this tropomyosin are going to cover your okay, chain of your actin. It's going to be covering like this. It is going to be covering like this, and this is your tropomyosin. Okay, this tropomyosin is going to be masking. Okay, this is going to mask here. So your myosins வந்து attach ஆகக்கூடிய இந்த ஏரியாவை ஓகே ஆக்டின் மேல இந்த ட்ரோபோமயோசின் மாஸ்க் பண்ணி கவர் பண்ணி வைக்குது. அப்ப ஆக்டின் மயோசின் கிராஸ் பிரிட்ஜ் இஸ் மேட் பாசிபிள் by movement of tropomyosin away from your binding site. From your binding site, your tropomyosin has to move in order for a muscle contraction. In order to a muscle contraction to occur, this has to be there. Okay, now you know what is M line, Z line, everything. So what happens exactly? For a muscle to contract, your Z line has to come closer, the length of the H and I band has to decrease, and length of the A band is going to remain constant. Okay, even though there is an interaction between the actin and myosin, your A band will always remain constant. Your A band will always remain constant. Maybe your H band may decrease, your I band may decrease. Okay, your H band may decrease, your I band may decrease, but remember, your A band never decreases. Your A band never decreases because A band is thick filament. That means what I wanted to make it clear here is your thick filament that is going to be your H, so your thick filament that is going to myosin size does not decrease. Okay, size does not decrease. Your thin filament is the one that is going to move and overlap over your thick filament. Therefore, the length of H band and I band decreases. Your length of H band and I band decreases. Is it clear? Okay, anyway, we are going to see once again everything. Okay, let me can we answer? So you have this myosin. They are going to be composed of multiple myosin molecule. Each myosin molecule has 6 polypeptide. So the heavy chain wrap around each other to form the tail, forming the tail of myosin molecule. One end of each of the heavy chain is folded to form head. Each myosin molecule has 2 head within which enclose the light chain. Myosin head is going to be projecting from the body of myosin filament, cross bridges, interacts with actin, and there is going to be muscle contraction. Now tell me, now answer me. So what is going to be your thick filament made of? Tail of your myosin, whereas the contractile part of your myosin is going to be your head of myosin, your head of myosin. Okay, now again the central region of the myosin filaments are going to lack a cross bridges, and myosin head can function as a ATPase enzyme is present, which is going to give you the energy. I have given you what all I spoke about the actin, what all I spoke about the different parts of troponin. I have given you as a theory in this. Okay, so that you can read it slowly, you can understand it. Okay, wow. So now moving on towards the calcium. I want to talk this calcium separately because calcium is very, very important. They are the one that are going to be providing energy. They are the one that is going to be helping in coupling the excitation to contraction. Calcium ion are responsible for the muscle contraction. Calcium ion movement in the skeletal muscle are going to be regulated by a structure called as sarcotubular system, or sarcoplasmic reticulum is the one. Okay, so the sarcotubular system has 2 tubules. So you can see T tubules, isn't it? And you have a L tubule also. These 2 tubules, like T tubule வந்து பாத்தீங்கன்னா they are present on the sarcolemma, whereas L tubule is present inside your sarcoplasmic reticulum. Okay, ba? Inside your sarcoplasmic reticulum. Okay, so which stores and release your calcium, and they are called as terminal cisterns located at the junction of A and your I band. One T tubule is surrounded by 2 L tubule. Remember, one T tubule will be always surrounded by 2 L tubule. Clear? Okay, so the classical arrangement of this is called as a triad of your skeletal system. So the T tubule have a calcium channel. You call it as a dihydropyridine receptors. Dihydropyridine receptors. Where are these dihydropyridine receptors present? Can you see here? Okay, you have inside your T tubule, you're going to have certain channels, isn't it? You're going to have certain channels. So this is going to be your sarcoplasmic reticulum that is present between your T tubules. So T tubules are going to be a voltage gated calcium channel. They open at plus 40 millivolt. This one plus 4, this is going to open at the plus 40 millivolt. It is going to be opening at the plus 40 millivolt. Okay, so that is going to be during the peak of your depolarization. And there is this receptor is called as dihydropyridine receptor. This receptor is called as dihydropyridine receptor. What is this receptor called as? Dihydropyridine receptors. Okay, so these are the receptors which are going to open. The sarcoplasmic reticulum has got a mechanically gated calcium channel. This is going to have a mechanically gated calcium channel. So that is called as your dihydropyridine receptor, which is nothing but your dihydropyridine receptor. Okay, then you're going to have this dihydropyridine is called as ryanidine receptor. Okay, so remember, so this ryanidine receptor is present in the sarcoplasmic reticulum. So first you're going to have a T tubule. Understand this carefully. You're going to have a T tubule, transverse tubule. Inside this transverse tubule, from this place, transverse tubule வந்து பாத்தீங்கன்னா, okay, இதான் உங்க transverse tubule. இந்த transverse tubule குள்ள உங்களுக்கு வந்து பாத்தீங்கன்னா calcium channels இருக்கும். அந்த calcium channels open ஆகணும்னா, okay, அதாவது இந்த endல you have two ends. Can you listen carefully? So you have two ends. Okay, so you have, this is your T tubule. Okay, this is going to be your T tubule. Your T tubule is going to be like this. At this place you're going to have one receptor. At this place you're going to have other receptor. Okay, the receptor which is present in the T tubule, you call it as a dihydropyridine receptor, and the receptor which is present in the sarcoplasmic reticulum opening, that is called as ryanidine receptor. So T tubule irukadhu voltage gated calcium channel, and the one present in the calcium sarcoplasmic reticulum, that is going to be a ryanidine receptor, which is mechanically gated calcium channel. So what happens? This opening of a voltage gated calcium channel along with the mechanically gated calcium channel, both has to open. So this opening is called as a electromechanical coupling. This is called as electromechanical coupling, which is going to be playing an important role in the muscle contraction. Is it clear? Okay, so what is this dihydropyridine receptor? Certain drugs like your amlodipine, nifedipine, nicardipine, இது எல்லாமே வந்து நம்ம hypertension use பண்ற drugs. So these drugs are going to be acting as a calcium channel blockers. Which channel it is going to block? Voltage gated calcium channel. What is the name of that voltage gated calcium channel? Dihydropyridine receptor. Clear? Now you have the other one that is called as ryanidine receptor. These ryanidine receptors, where they are going to be helping, it is in malignant hyperthermia. So what is this malignant hyperthermia? This is an autosomal dominant disease due to the mutation of this ryanidine receptor. So what happens? Because of the mutation of this area, there is going to be excess calcium from the sarcoplasmic reticulum that leads to excess muscle contraction. And when muscle is contracted excessively, there is a heat production. This heat production represents as a hyperthermia. How you are going to treat this? By blocking this ryanidine receptor. What are the drugs that are going to block your ryanidine receptor? Dantrolene. Dantrolene is a drug that blocks the ryanidine receptor. So I have told about two receptors, dihydropyridine and then ryanidine. Okay, so what is a drug that blocks your dihydropyridine? Dihydropyridine is blocked by amlodipine. Okay, and you have this nifedipine, you have this amlodipine, etc. And ryanidine receptor is blocked by dantrolene. You should never forget this, because this is what you're going to read in the next pharmacology next year. That is also going to be dealing with the same physiology. When you understand physiology, pharmacology is easy, pathology is easy, medicine is easy, everything is easy. Okay, now what happens? Now you understood how there is going to be a muscle contraction. So opening of the voltage gated channel is called as electromechanical coupling. Electromechanical coupling. Right. Okay, now since you understood this, Now, since you understood this entire process, now you have to understand how there is a excitation contraction. How there is going to be excitation contraction. This is your relaxed state. You have the myosin head. You are going to be attached with the, okay, your myosin head is going to be present over your actin, over your thin filament. So this is your thick filament. This is your thin filament. So this is going to be your thick and this is going to be your thin. This interaction, but who is preventing this interaction? Tropomyosin, that blocks your binding site on that G-actin. Okay, that is going to blocking your binding of your G-actin. Now, what happens is, when there is going to be action potential, there is going to be a calcium release. Remember, there is going to be calcium release. This calcium enter into the cytoplasm, and this calcium is going to be first binding with, this calcium is going to be first binding with troponin. Which troponin? Trop C. So, trop C la poi first okarudhu. Trop C la when it sits, that is going to activate your trop C. Okay, now activation of trop C, what happens? There is going to be a activation of trop T. Okay, what is your trop T? Tropomyosin. So this trop T is, okay, making your tropomyosin. This trop T is making your tropomyosin to cover your muscle binding site. Now, when this trop T is acting, this is going to be activating tropomyosin. Tropomyosin. When tropomyosin is going to be acted, that is going to be moving. That is going to be moving from the myosin binding side. Therefore, what happens? Your actin myosin yet is going to combine with the actin, and this is going to form contraction. Very easy process, isn't it? So this is very easy process when you understand the concept. Now, how is this calcium spilled out? That you need to understand, right? So when there is a sarcoplasmic reticulum, where here is going to be more calcium. Whenever there is going to be a plus 40 millivolt, it opens the voltage gated calcium channels of your dihydropyridine receptor. This in turn opens your ryanodine receptor. The calcium that is going to be stored inside your sarcoplasmic reticulum is spilled into your cytoplasm of your muscle fibers. That is going to combine with the trop C, then trop T, then tropomyosin. Then it's going to moving away, exposing your binding site. Actin myosin interaction takes place. That's all. Okay, so that is the entire idea. Okay, that is the entire idea. Myosin actin cross bridge is formed. Okay, there is going to be myosin actin cross bridge formation. Once there is going to be cross bridge formation, there is going to be power stroke. I call it as power stroke. What is a power stroke? I'll tell you. Okay, so power stroke is nothing but there is going to be energy released. There is going to be energy release. Okay, ADP is released. There is a profound changes that is going to take place in the intramolecular level between the head and arm. Your head is going to tilt towards your arm. Can you see a head is going to be tilting towards your arm? This tilt is going to cause movement of your muscle fiber. This is going to cause contraction. This is going to cause contraction. Clear? So this is going to cause contraction. So there is going to be binding. Now myosin is free to interact with actin. Upon the formation of cross bridge, what happens? ADP is released. This ADP is going to cause a conformational change. Conformational change in the myosin head that moves the thin filament relatively to the thick filament, comprising of cross bridge power stroke. This sliding of microfilament is the basis of your sliding filament theory. Once there is a power stroke, the ATP is going to again bind with the myosin head. So this ATP is going to be binding with the myosin head. Remember, until your tropomyosin and ADP is present, it is not going to cause actin myosin interaction. Only when ATPase is going to act. So how this ADP is released? By the action of ATPase enzyme. This ATPase enzyme, what it does is, it is going to be the first. So because of this calcium influx, because of all these things, what happens is, there is going to be ATPase. This ATPase is the one that produces ADP. Because of the production of ADP and the energy, okay, phosphate, the energy, that energy is going to cause your power stroke. Once the power stroke ends, your ATP again, new ATP comes, they bind to the myosin head, and whenever ATP binds with myosin head, it gets detached from your actin. So the actin and the detach ஆயிடுது. So myosin head has a ATPase activity. The ATPase role, the ATPase role, we call it as a relaxation phase. So what happens is, up to this point, you saw that there is a myosin that is going to be binding with actin. So in order to muscle to relax, in order for a muscle to relax, the calcium must go back into the sarcoplasmic reticulum. But what happens is, the channels which used for the calcium to come out of your sarcoplasmic reticulum, that is going to be your ryanodine receptor. You cannot use the same ryanodine receptor. Okay, so you cannot use the same ryanodine receptor because the calcium concentration is more inside your sarcoplasmic reticulum. Therefore, you need a proper active transport mechanism. Your ryanodine all are going to be voltage gated, mechanical gated receptors that will help you for the flow of ion from a higher concentration to a lower concentration. But here you have to push from the lower concentration inside the higher concentration. For that you need energy. So this entire ATP to ADP conversion, everything after the power stroke gets completed, you need to have a proper mechanism that is called as SERCA. That is called as SERCA. That is what we are going to see here. SERCA. What is the SERCA? It is sarcoplasmic endoplasmic reticulum calcium ATPase. Sarcoplasmic, come on, write along with me. Sarcoplasmic endoplasmic reticulum calcium ATPase. This is going to be helping. This is going to be the one that is going to be helping in the entire process. This ATPase, what it will be doing? This ATPase are going to, what happens? If they are going to, it's going to be a pump, isn't it? So this ATP is going to be quickly bind with the exposed site of the myosin. So what happens? First, for an initial thing to occur, your ATP is broken down to form ADP and phosphate. Okay, once there is going to be flexion of your head of your myosin, okay, once there is going to be flexion of your head of your myosin, what happens is, there is going to be a, normally what happens, once there is going to be head of your myosin is going to be flex. Okay, once there is going myosin actin interaction, here you are going to have hydrolysis. You are going to have hydrolysis where ATP is going to be converted into ADP plus phosphate. Okay, this ADP is released. Now you have the exposure. Since there is an exposure, there is going to be this phosphate is going to give a power stroke. That is what I told you earlier. Okay, this is going to cause the flexion of your tail. Then the thin filament is pulled inside your sarcomere. The length of the muscle is shortened. Therefore, the I band is going to be shortened. That is what we saw earlier. So your Z line comes together, your I band, H band shortens, and the length of A band remains constant. Now what happens? You're going to have ADP plus PI, isn't it? So this ADP plus PI in the presence of SERCA, it is going to be again converted into ATP, and this ATP, what happens? They quickly bind to the free exposed site of the myosin. That is, it is going to detach the head of your myosin from your actin. Once there is a detachment of ATP from your actin, this ATP comes back, and this is going to quickly cause inhibition of the skeletal muscle contraction. So this entire thing, you call it as a power stroke. So the sliding during the muscle contraction occurs when the myosin head firmly attaches with the actin, bend at the junction of head with the neck and arm, and then detach. This is going to be caused by power stroke depending on hydrolysis of your ATP. Once your ATP hydrolyzed, the phosphate is released, causing recocking of the myosin head and completing the cycle. Okay, this is going to complete the cycle. It is free once again when this hydrolysis occurs, when the calcium is available. Once again, ATP is going to be broken down to ADP plus phosphate. Once again, the ADP is going to come. Then once again, what happens? There's actin myosin interaction. The entire contraction relaxation is going to get continued. Clear? So what happens in between? Your I band is going to be reduced. Your A band is going to remain static. The thin filament encroaches your H zone and touches your M line. The H zone practically disappears. At the point where there is going to be actin myosin interaction, your H zone is going to disappear. So all these things, why I am telling is, this can be asked as a MCQ. Okay, so your H zone disappears. So I band reduces, A band remains constant, H zone is going to disappear. All these points can be asked as a MCQ. So your thick filament brought into the sarcomere, two sides of your thin filament comes very close, H zone completely disappears. When there is a relaxation, your head of the myosin is going to be attached with ATP. When there is a contraction, ATP is going to be hydrolyzed to ADP and phosphate. So to relax, so the important thing here is, in order to have a relaxation, you need to have a new ATP molecule. So one thing you need to be very clear is, in order for a muscle to relax, you need a new ATP molecule. This ATP binding is very much necessary. So once the contraction is over, calcium ions are immediately pumped back into sarcoplasmic reticulum. Who is going to pump back into sarcoplasmic reticulum? This is going to be done by Cerca. This is going to be done by Cerca. So once calcium concentration outside the sarcoplasmic reticulum is lowered, chemical interaction between actin myosin is going to be reduced, and your muscle will be relaxing. So ATP is used as an energy source for everything. ATP is the only substance in the muscle used both for contraction as well as for relaxation. So both for contraction and relaxation, you need new ATP, because your ATPase enzyme is just an enzyme. For that to be done, you need new ATP molecule. So for two places, ATP molecule is required. One, for completing your contraction. To stop your contraction, you need ATP. To stop contraction, you need ATP. That is the interaction between your actin and your myosin. Similarly, you need ATP for your Cerca to act. Okay, that's all. So this is what you need to understand. I hope you all understand this. Okay, so this will be the first part of your muscle physiology. After completing this first part of muscle physiology, in the part 2, we will be discussing about the mechanisms, or you will be discussing about the different properties of your muscle. Then we will discuss about the smooth muscle. We will be discussing about the neuromuscular junction. Okay, then we will discuss about the two important diseases, myasthenia gravis and your Lambert-Eaton syndrome, in the next session. Thank you so much.